Our next speaker is Marie-Hélène Masse. She's a respiratory therapist by training and now works as a project manager and research coordinator. For those of us who do clinical trials, you know that the main success of the trial really depends on a high quality and really hardworking project manager, for which Marie-Hélène has led many trials across Canada. And today she's going to share some of her advice and lessons learned. So, Ms. Masse.
So thank you for the opportunity to present at this meeting. So as Dr. Boyd said, I'm a respiratory therapist by training, and I did a master in health research methodology. So I've been involved in sepsis trials in the past 13 years as a research coordinator, but also as a project leader. So I have experience in both roles. And I'm now the full-time project manager of the Canadian Clinical Research Network. So today I will talk about the current landscape. I will also share how I think we should address the challenges, and I will share one example of successful collaboration. So, I will start with a typical scenario for a research coordinator working in the ICU. So we are Friday night, September 8th, it's 7 p.m. and a 75-year-old woman is admitted to the ICU. She had a cardiac arrest at home, discovered by her neighbor. She's unconscious, her mean arterial pressure is at 50 millimeters of mercury, and her temperature is 40 Celsius degrees. So at 8 p.m., the suspected diagnosis by the physician is a pneumonia with severe hypotension and desaturation. She's on mechanical ventilation and receiving vasopressors. They did some x-rays and laboratory sampling. And few minutes later, they confirmed the diagnosis as a septic shock. So the physician calls the research team to let them know that there is a potential candidate for sepsis trials. So at this site, there are 2 sepsis RCTs, 1 observational study about sepsis and a biobank that is approved by the REB. So the first sepsis RCT has an eligibility window of 6 hours from ICU admission. Deferred consent is approved by the REB, and the pharmacy is required to prepare the investigational product. The second sepsis RCT has a larger, more time to include the patient, so 48 hours from ICU admission. Regular consent and substitute decision maker consent are approved. Pharmacy is also required. There are blood sampling on day 1, day 3, and day 7. The third study is an observational study, so only data collection is required. And the last one is a biobank, so the patient must be included during the time he is in the ICU. So regular consent is approved, and blood samples are drawn on day 1, day 3, and day 7. So for the research coordinator, the challenges are, from my point of view, the following. So the eligibility window. So since the patient is admitted in the ICU, the clock starts. So 6 hours mean having the diagnosis confirmed by the physician. The physician will have to talk to the family before the research team approach the family for consent. So 6 hours to randomize a patient might be a challenge. Also, as I told you before, it was out of business hours, so the RC is probably at home. If it's in a large city, getting on-site will be also a challenge to do that in like an hour. So that's an important challenge for the RC. Co-enrollment, so sometimes the REB will allow a maximum of 2 studies in which the patient can be included. So that makes the RC and the PI to do some choices. Of business hours admission, I think that's the biggest challenge. So of business hours means that the pharmacy might be closed. So a lot of sites have the pharmacy available until 4 p.m. This also means that the RC will need to confirm eligibility with a PI and a co-PI or a co-PI, so they must also be available during off-business hours. This might also means that the PI will need some extra funds to cover the on-call time of the RC. At some site, the samples must be processed by a specific team, so also they must be available to process the samples. So this is an important challenge for the RC, but from my experience at our site, we include at least 50% of our patients during off-business hours. So if you would like to work on one challenge, I think that's the one to work on if you want to keep your recruitment rate high at your site. Consent models, that's, as you know, if deferred consent is allowed in a study, that might facilitate recruitment, especially during off-business hours, during weekends, during night. And even if the eligibility window is short, the family might not be adequate to approach the family during the first hours of admission. And the contact with family, so sometimes it takes hours, days to find some family to the patient, so the RC must be really in contact with the physician before approaching the patient or the family. On the site level, so the challenges for the local site, having many trials means many RAB submission, many agreements to deal with limited human resources, especially during the pandemic. So nurses, respiratory therapists were asked to go to the bedside during the pandemic. So that was a challenge also. So this also means decrease in capacity to start new studies, a decrease of the time they spent in the ICU for screening and randomization. So this results in a decrease in research capacity at this site. On the challenges of the method center, so the method center we mean where the PI and the project leader will manage the clinical trials. So having many trials also means many RAB submission agreements and limited resources, but this increased the delays for site activation. As I said, decrease the screening and randomization at the sites, decrease the recruitment rate, and increase the period of recruitment to reach the sample size. And if you extend the period of recruitment, you increase your cost of your study because you need to pay the project leader longer. So this is also a decrease in research capacity. Now, how to address the challenges? So the first step to increase research capacity is to monitor research, to quantify and describe where research is occurring. So the Canadian Clinical Research Network is led by François Lamontagne and Michel Coe, who will present in a few minutes. So the vision of the Canadian Clinical Research Network is that conducting high quality and efficient clinical research is essential for improving the healthcare available to Canadians. It is also a vector for innovation and discovery. The mission is to improve clinical research and coordination across Canada with the objective of increasing the production of clinical evidence that will inform healthcare decisions and to increase the wealth of data and biological specimens available for translational research. So far, we have collected information about acute care sites, so hospitals across Canada, and information about clinical studies. We have included this in a database, and we are able to show where could research occur in Canada and where actually does research occur in Canada. So the blue dots are hospitals where no patient are included in the sepsis trials that we have in our database. And the red dots are the sites where the patients are included in sepsis trials. So you see that there is potential to increase research capacity. So I'm coming back to that slide with the list of studies that are approved at this site. And if I had one question today, I would ask you, in which trial would you include this patient? So if I answer this question, my answer would be this one. So I would choose to include the patient in the second one, so the sepsis RCT, with samples on day 1, day 3, and day 7, and the biobank, for which the samples are the same time points. So the RC will go and get the samples, so only like combine the samples together at the same time points. The consent will be done at the same time, so it won't be much more work for the RC. They will combine the procedure together. There will be patient in the intervention arm, but also in the placebo, and some samples will be available later to answer future research questions. So this leads to talk about biobanks. So by definition, it's combining data and biological specimens. So we say standalone biobanks when we collect and store data to answer future research questions. But when we collect specimen within a study to answer a specified question specific to the study, and that we also get a consent to biobank the rest of the samples after the analysis are done, and we combine this to the data that are collected within the study, we obtain a nested biobank. In other words, combining clinical study data with the clinical specimen collected with the study, with the consent of biobanking the specimens, we can answer future research questions and increase capacity with the same resources. So one example of successful collaboration that arose from the CCRN is the following. So the PI of the Ovation 65 trial, which is François Lamontagne, and the PI of the ARB Corona trial, which is Dr. Russell, Jim Russell, they collaborate together because Dr. Russell needed some specimen from septic patient to compare with COVID-19 patient, and François had collected some biospecimen from the Ovation 65 trial and obtained consent for biobanking. So after he did his analysis for the Ovation 65 trial, there were specimens available to be shared with Dr. Russell. So this is an example where a collaboration, they save some time and some money, and the new findings arose from this collaboration. So maybe you would like to know if researchers have some samples available to be shared. So you can find this information on the CCRN website. So there is a specific page where you can find the sample information collected by each study and the variables that are collected. You can apply some filters to these tables. Like you can select only pediatric studies or sepsis trials, and you can extract these tables in Excel format, and you can then contact the researchers to start new collaboration. You can find information so far on 4 standalone biobanks, 14 nested biobanks. There is information about the consent for genetics, the time points that the samples are collected, and the type of specimen that are collected. So in summary, I think we should encourage researchers to design sepsis trial with a nested biobank, and also encourage researchers to collaborate with those who have specimen available, because everyone will save money. We will make life easier for RCs. The patient contribution will still be the same, but will have more impact. Everyone will save time. Institutions, funders, and governments will benefit with an increased productivity. And one last thing, if you are a PI or project leader of a study that is not already included in the CCRN database, please contact me and we will work together to include it as soon as possible. Thank you.
Marie-Hélène, how do we get more community intensive care units engaged in research in general, but sepsis specifically?
That's a good question. I think sometimes community have the potential to, so as you saw, there are a lot of blue dots, and maybe Michelle will share a few information about community versus academic side, but there are a lot more community sites than academic sites, and that's where I think we should help those teams, like as mentors, or maybe share our experience, because I think that's feasible, and there are few community sites that have a great recruitment rate across Canada, and maybe they can share also their experience and how they found solution to, and from my experience, at many sites have a specific team that will process the lab, but also, and at our site, even if we are a university site, that's our research team who process the samples. And we have hired some students to be available during our business hours, so they can, that's an opportunity for them to put this in their CV, but that's also cheaper for the PI to have everyone available during our business hours. So that's one potential solution. Thank you.